Gen 1 is a novel IL-12 immunotherapy designed with Imunon's proprietary TheraPlas technology. When delivered to a tumor site, Gen 1 may potentiate both innate and acquired immune responses against cancer. Advanced epithelial ovarian carcinoma is a lethal cancer in which the complete surgical resection of tumors is difficult to achieve, and recurrences are common. Chemotherapeutic regimens have not been proven curative in newly diagnosed or recurrent disease. They do, however, induce severe toxicities. Novel immunotherapeutic approaches are being investigated in advanced epithelial ovarian carcinoma. While traditional chemotherapy kills fast-growing cancer cells, immunotherapy enhances the local and systemic immune response against tumors, potentially offering a more targeted and possibly less toxic approach to cancer treatment. The cytokine interleukin-12, IL-12, can induce anti-cancer immunity through multiple mechanisms. Firstly, IL-12 promotes cellular production of the potent immune mediator interferon gamma. Secondly, IL-12 stimulates the proliferation of CD8-positive T cells and natural killer cells and their cytotoxic activity against the tumor. Thirdly, it shifts the differentiation of naive CD4-positive T cells towards a Th1 phenotype, further enhancing the immune response. Fourthly, regulatory T cells that suppress immune responses may be inhibited by IL-12. Finally, interferon gamma promotes the expression of anti-angiogenic molecules, halting the growth of new blood vessels that supply oxygen to the tumor. Gen 1 consists of a human IL-12 plasmid formulated with a non-viral synthetic DNA delivery system, polyethylene glycol polyethylene imine cholesterol, PPC. The PPC polymer condenses the plasmid DNA, forming nanoparticles. The lyophilized nanoparticles are simply reconstituted with sterile water at the patient's bedside, and the solution is infused directly into the local tumor environment in the abdomen via a catheter. The three components of PPC play different roles in bringing plasmids to their target cells. Polyethylene imine, PEI, condenses the DNA strands into nanoparticles by electrostatic interaction. Polyethylene glycol, PEG, improves in vivo stability and protects the plasmid from clearance by serum proteins. Cholesterol is a biocompatible moiety that enhances trafficking across cell membranes. The nanoparticles can thus become endocytosed by cancer and non-cancer cells in the tumor microenvironment. Within the cell, polyethylene imine, PEI, facilitates lysis of the endosome, allowing the naked DNA plasmid to emerge and enter into the nucleus. The IL-12 gene it encodes can now be expressed by the cellular machinery. Transfected cells are able to produce sustained concentrations of IL-12 protein in the vicinity of the tumor. Gen 1 thus provides for a local and durable elevation of IL-12 at the tumor site, with exposure that persists for several days after a single treatment. The secreted IL-12 does not accumulate locally. This may minimize toxicity by avoiding spillover into the systemic circulation. Durable concentrations of IL-12 may lead to prolonged infiltration of immune cells in the tumor environment, enhancing the local immune response against cancer. The safety, biological activity, and anti-tumor activity of Gen 1 have been demonstrated in Phase 1 and Phase 2 clinical trials in ovarian cancer patients. Gen 1 activity has also been observed in multiple tumor types in preclinical studies including glioblastoma, intraperitoneal colorectal cancer, and intraperitoneal pancreatic cancer.